The Venus flytrap, also referred to as Venus's flytrap or Venus flytrap, Dionaea muscipula, is a carnivorous plant native to subtropical wetlands on the east coast of the United States in North Carolina and South Carolina. It catches its prey, chiefly insects and arachnids, with a trapping structure formed by the terminal portion of each of the plant's leaves, which is triggered by tiny hairs on their inner surfaces. When an insect or spider crawling along the leaves contacts a hair, the trap closes if a different hair is contacted within 20 seconds of the first strike. The requirement of redundant triggering in this mechanism serves as a safeguard against wasting energy by trapping objects with no nutritional value. Dionaea is a monotypic genus closely related to the waterwheel plant and sundews, all of which belong to the family Droseraceae. Description: the Venus flytrap is a small plant whose structure can be described as a rosette of 4 to 7 leaves, which arise from a short subterranean stem that is actually a bulb-like object. Each stem reaches a maximum size of about 3 to 10 centimeters. Depending on the time of year, longer leaves with robust straps are usually formed after flowering. Flytraps that have more than 7 leaves are colonies formed by rosettes that have divided beneath the ground. The leaf blade is divided into 2 regions, a flat, heart-shaped photosynthesis-capable petiole, and a pair of terminal lobes hinged at the midrib, forming the trap which is the true leaf. The upper surface of these lobes contains red anthocyanin pigments and its edges secrete mucilage. The lobes exhibit rapid plant movements, snapping shut when stimulated by prey. The trapping mechanism is tripped when prey contacts one of the 3 hair-like trichomes that are found on the upper surface of each of the lobes. The mechanism is so highly specialized that it can distinguish between living prey and non-prey stimuli, such as falling raindrops. 2 trigger hairs must be touched in succession within 20 seconds of each other or 1 hair touched twice in rapid succession, whereupon the lobes of the trap will snap shut, typically in about 1/10 of a second. 6. The edges of the lobes are fringed by stiff hair-like protrusions or cilia, which mesh together and prevent large prey from escaping. These protrusions, in the trigger hairs, also known as sensitive hairs, are likely homologous with the tentacles found in this plant's close relatives, the sundews. Scientists have concluded that the snap trap evolved from a flypaper trap similar to that of Drosera. The holes in the meshwork allow small prey to escape, presumably because the benefit that would be obtained from them would be less than the cost of digesting them. If the prey is too small and escapes, the trap will usually reopen within 12 hours. If the prey moves around in the trap, it tightens and digestion begins more quickly. Speed of closing can vary depending on the amount of humidity, light, size of prey, and general growing conditions. The speed with which traps close can be used as an indicator of a plant's general health. Venus flytraps are not as humidity dependent as are some other carnivorous plants, such as nepenthes, cephalotos, most Helianthera, and some Drosera. The Venus flytrap exhibits variations in petiole shape and length and whether the leaf flies flat on the ground or extends up at an angle of about 40 to 60 degrees. The 4 major forms are, typica, the most common, with broad decumbent petioles; erecta, with leaves at a 45-degree angle; linearis, with narrow petioles and leaves at 45 degrees. And filiformis, with extremely narrow or linear petioles. Except for filiformis, all of these can be stages in leaf production of any plant depending on season, decumbent in summer versus short versus semi-erect in spring, length of photoperiod, long petioles in spring versus short in summer, and intensity of light. Wide petioles in low light intensity versus narrow in brighter light. When grown from seed, plants take around 4 to 5 years to reach maturity and will live for 20 to 30 years if cultivated in the right conditions. Etymology: the plant's common name refers to Venus, the Roman goddess of love. The genus name, Diania, daughter of Dion, refers to the Greek goddess Aphrodite, while the species name, Muscipula, is Latin for mousetrap. Historically, the plant was also known by the slang term tipitywitchet or tipitty-twitchet, possibly an oblique reference to the plant's resemblance to human female genitalia. Carnivory, prey selectivity: most carnivorous plants selectively feed on specific prey. This selection is due to the available prey and the type of trap used by the organism. With the Venus flytrap, prey is limited to beetles, spiders and other crawling arthropods. In fact, the Diania diet is 33% ants, 30% spiders, 10% beetles, and 10% grasshoppers, with fewer than 5% flying insects. Given that Diania evolved from an ancestral form of Drosera, carnivorous plants that use a sticky trap instead of a snap trap, the reason for this evolutionary branching becomes clear. Whilst Drosera consume smaller, aerial insects, Diania consume larger terrestrial bugs. From these larger bugs, Diania are able to extract more nutrients. This gives Diania an evolutionary advantage over their ancestral sticky trap form. Mechanism of trapping: the Venus flytrap is one of a very small group of plants capable of rapid movement, such as Mimosa pudica, the telegraph plant, sundews and bladderworts. The mechanism by which the trap snaps shut involves a complex interaction between elasticity, trigger and growth. The trap only shuts when the trigger hair is stimulated twice. This is to avoid inadvertent triggering of the mechanism by dust and other wind-borne debris. In the open, untripped state, the lobes are convex, bend outwards, but in the closed state, the lobes are concave, forming a cavity. It is the rapid flipping of the stable state that closes the trap, but the mechanism by which this occurs is still poorly understood. When the trigger hairs are stimulated, an action potential, mostly involving calcium ions, see calcium in biology, is generated, which propagates across the lobes and stimulates cells in the lobes and in the midrib between them. It is hypothesized that there is a threshold of ion buildup for the Venus flytrap to react to stimulation. After closing, the flytrap counts additional stimulations of the trigger hairs, to 5 total, to start the production of digesting enzymes. The acid growth theory states that individual cells in the outer layers of the lobes and midrib rapidly move 1H+ hydrogen ions into their cell walls, lowering the pH and loosening the extracellular components, which allows them to swell rapidly by osmosis, thus elongating and changing the shape of the trap lobe. Alternatively, cells in the inner layers of the lobes and midrib may rapidly secrete other ions, allowing water to follow by osmosis, and the cells to collapse. Both of these mechanisms may play a role and have some experimental evidence to support them. Digestion: if the prey is unable to escape, it will continue to stimulate the inner surface of the lobes, and this causes a further growth response that forces the edges of the lobes together, eventually sealing the trap hermetically and forming a stomach in which digestion occurs. Digestion is catalyzed by enzymes secreted by glands in the lobes. Oxidative protein modification is likely to be a predigestive mechanism used by Diania muscipula. Aqueous leaf extracts have been found to contain quinones such as the naphthquinone plumbago that couples to different NAD-dependent diaphoraces to produce superoxide and hydrogen peroxide upon hot oxidation. Such oxidative modification could rupture animal cell membranes. Plumbago is known to induce apoptosis, associated with the regulation of the BCL2 family of proteins. When the DNA extracts were pre-incubated with diaphoraces and NAD in the presence of serum albumin, subsequent tryptic digestion of SA was facilitated. Since the secretory glands of Droseraceae contain proteases and possibly other degradative enzymes, it may be that the presence of oxygen-activating redox cofactors function as extracellular predigestive oxidants to render membrane-bound proteins of the prey, insects, more susceptible to proteolytic attacks. Digestion takes about 10 days, after which the prey is reduced to a husk of chitin. The trap then reopens, and is ready for reuse. Evolution. The carnivorous diet is a very specialized form of foliar feeding, and is an adaptation found in several plants that grow in nutrient-poor soil. Carnivorous traps were naturally selected to allow these organisms to compensate for the nutrient deficiencies of their harsh environments by supplementing ordinary photosynthate with animal proteins. The snap trap mechanism characteristic of Dyneia is shared with only one other carnivorous plant genus, Aldrovanda. For most of the 20th century, this relationship was thought to be coincidental, more precisely an example of convergent evolution. Some phylogenetic studies even suggested that the closest living relatives of Aldrovanda were the sundews. It was not until 2002 that a molecular evolutionary study, by analyzing combined nuclear and chloroplast DNA sequences, indicated that Dyneia and Aldrovanda were closely related and that the snap trap mechanism evolved only once in a common ancestor of the two genera. A 2009 study presented evidence for the evolution of snap traps of Dyneia and Aldrovanda from a flypaper trap like Drosera regia, based on molecular data. The molecular and physiological data imply that Dyneia and Aldrovanda snap traps evolved from the flypaper traps of a common ancestor with Drosera. Pre-adaptations to the evolution of snap traps were identified in several species of Drosera, such as rapid leaf and tentacle movement. The model proposes that plant carnivory by snap trap evolved from the flypaper traps, driven by increasing prey size. Bigger prey provides greater nutritional value, but large insects can easily escape the sticky mucilage of flypaper traps. The evolution of snap traps would therefore prevent escape and kleptoparasitism, theft of prey captured by the plant before it can derive benefit from it, and would also permit a more complete digestion. Proposed evolutionary history. Carnivorous plants are generally herbs, and their traps the result of primary growth. They generally do not form readily fossilizable structures such as thick bark or wood. As a result, there is no fossil evidence of the steps that might link Dyneia and Aldrovanda, or either genus with their common ancestor, Drosera. Nevertheless, it is possible to infer an evolutionary history based on phylogenetic studies of both genera. Researchers have proposed a series of steps that would ultimately result in the complex snap trap mechanism. Larger insects usually walk over the plant, instead of flying to it, and are more likely to break free from sticky glands alone. Therefore, a plant with wider leaves, like Drosera falconeri, must have adapted to move the trap and its stalks in directions that maximized its chance of capturing and retaining such prey, in this particular case, longitudinally. Once adequately ramped, escape would be more difficult. Evolutionary pressure then selected for plants with shorter response time, in a manner similar to Drosera bermanii or Drosera glandulae era. The faster the closing, the less reliant on the flypaper model the plant would be. As the trap became more and more active, the energy required to wrap the prey increased. Plants that could somehow differentiate between actual insects and random detritus, rind droplets would have an advantage, thus explaining the specialization of inner tentacles into trigger hairs. Ultimately, as the plant relied more on closing around the insect rather than gluing them to the leaf surface, the tentacles so evident in Drosera would lose their original function altogether, becoming the teeth and trigger hairs, an example of natural selection utilizing pre-existing structures for new functions. Completing the transition, the plant eventually developed the depressed digestive glands found inside the trap, rather than using the dews in the stalks, further differentiating it from genus Drosera. Habitat: the Venus flytrap is found in nitrogen and phosphorus-poor environments, such as bogs and wet savannas. Small in stature and slow-growing, the Venus flytrap tolerates fire well, and depends on periodic burning to suppress its competition. Fire suppression threatens its future in the wild. It survives in wet sandy and peaty soils. Although it has been successfully transplanted and grown in many locales around the world, it is native only to the coastal bogs of North and South Carolina in the United States, specifically within a 60-mile radius of Wilmington, North Carolina. One such place is North Carolina's Green Swamp. There also appears to be a naturalized population of Venus flytraps in northern Florida as well as an introduced population in western Washington. The nutritional poverty of the soil is the reason that the plant relies on such elaborate traps. Insect prey provide the nitrogen for protein formation that the soil cannot. The Venus flytrap is not a tropical plant and can tolerate mild winters. In fact, Venus flytraps that do not go through a period of winter dormancy will weaken and die after a period of time.